তাহলে স্টার্ট করছি।
আমরা লাইভে আছি,
ওকে 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 ওকে। A very good afternoon to our principal, respected Dr. Prashant Bhattacharya, IQAC coordinator, Professor Tanmoy Bandhopadhyay, respected speakers, Professor RAJIB Bandhopadhyay, Professor UGC Center for Advanced Study, Department of Botany, Burdwan University. Dr. SAYAK GANGULI, Assistant Professor, Department of Biotechnology, St. Xavier's College, Kolkata. All other distinct viewers, colleagues, friends, and most importantly, my dear students. I cordially welcome you all in today's one-day popular talk entitled Glimpses of Bioinformatics for the Beginners, jointly organized by the Department of Botany and Internal Quality Assurance Cell, Rabindra Mahavidyalaya, Chapadam. The era of information technology has enriched the knowledge of different fields of basic and applied biology. Accumulation, storage, and proper dissemination of biological data on the whole have led to this leading edge technology known as the bioinformatics. It is often synonymously called computational biology. However, both these science branches have very subtle but distinct differences. Computational biology deals mainly with the data accumulation and organization using the computational algorithms, while bioinformatics moves beyond data management to the analysis and interpretation of the biological systems. Bioinformatics is also the connector of interdisciplinary science that combines biology with the computer science, mathematics, information engineering, and also statistics. Although not as a replacement, but always as an important complement, bioinformatics has augmented experimental biology. In fact, it is now considered as an important and indispensable field of science in drug discovery and many allied branches. We are very happy to have with us two distinguished resource persons who will enlighten our young students who are likely to enter into the knowledge world of bioinformatics as beginners. But before hearing from our speakers, I would like to request our principal, sir, to say a few words for our students and deliver his inspiring inaugural speech. Sir, please.
Thank you, Shelly. As the hostess of this particular program, as also other teachers are present, I don't know whether my speech, this inaugural one or not, is going to be an inspiring one, but definitely I'm inspired as a hearer, as a listener, and as a participant in this particular field. As a kind of a non-starter, and as a man who doesn't belong to this particular discipline of study, the kind of information I gather right now, or the impression that presently is formed, is of a fast developing field of study that is interdependent and what we call as interdisciplinary. We are happy to have among us two wonderful and well-known teachers from two very famous universities in and around Kolkata. Burdwan is not really around Kolkata. It's a kind of distant place, but not so far away as from Kolkata as not to be a part of the known field of the kind of people that we continually interact with, and particularly because Burdwan happens to be our affiliating university. So it is only natural that Professor Bandhopadhyay is part of this program, and he's our first speaker. So as principal of my college, I heartily welcome you, sir, to be the first speaker. And I have come to know that you are in a bit of a hurry, so there is a need to start the program right away. And also, I extend my hearty welcome to the other speaker, Professor SAYAK Ganguli, who is from St. Xavier's College and University. He belongs to the Department of Biotechnology. So it is only natural that we have two very eminent speakers with us, as we also have our teachers of our Department of Botany and the IQAC coordinator, as also a student. So this is a kind of a learning step for all of us, especially for my dear students. And by the way, at the very beginning, when the program hasn't yet started officially, as the slides were being shown on the screen, I do have this kind of an idea that Professor Bandopadhyay is particularly concentrating on the use of mobile phone as effectively as possible for studying this newly developing area in botany and other related fields of biological studies. So that is going to be very interesting, because the smartphone almost has become a feature of our modern life. And not only that, for most of our students, I cannot always guarantee a PC or a laptop or any other kind of more expensive or sophisticated device for him or her, but at least rather inexpensive smartphone is not only a must, but is quite available, if not for all, but at least for most of them. So he has rightly, I think, has chosen to make maximum use of this particular device, which is also known for a lot of abuse as well. So let us see what awaits us in this particular kind of presentation that is going to make for us. And without much delay, I give it over to the hostess to take us to the next level of the proceeding. And thank you very much for coming and for enlightening us, sir.
Thank you very much, sir, for your motivational words. Now, I would take the opportunity to invite our IQAC coordinator, Professor Tanmoy Bandopadhyay, to say a few valuable words in view of today's popular talk on bioinformatics. Sir, please.
Hello.
Yes, sir.
শোনা যাচ্ছে?
হ্যাঁ স্যার, শোনা যাচ্ছে।
Thank you, Dr. Selim Madam. আমি নমস্কার জানাই সকলকে যারা আজকে উপস্থিত হয়েছেন আমাদের এই ওয়েবিনারে। আমি মাননীয় অধ্যক্ষ মহাশয়কে ধন্যবাদ জানাই যে অনবরত উনি এরকম প্রোগ্রাম সকলকে ইন্সপায়ার করছেন, প্রত্যেকটা ডিপার্টমেন্টকে উনি বজায় রাখছেন যে যাতে একটা ব্যালেন্স করে সমস্ত প্রোগ্রামগুলো চলতে থাকে। আজকে আমি আরো আপ্লুত যে আমাদের একটা ভালো ডিপার্টমেন্ট, বোটানি ডিপার্টমেন্ট আমাদের কলেজের অত্যন্ত গুরুত্বপূর্ণ একটা ভূমিকা পালন করে, এবং বোটানি ডিপার্টমেন্ট প্রত্যেকটা টিচার, নন টিচিং স্টাফ সকলেই, তো তাদের সাহায্যে এরকম একটা পপুলার টক, যার টপিকটা হচ্ছে গ্লিমসেস অফ বায়োইনফরম্যাটিক্স ফর দ্য বিগিনার্স, এরকম একটা সুন্দর টপিক চুজ করার জন্য, যেটা অবশ্যই আমাদের ছাত্রছাত্রীদের ভীষণভাবে উপকার করবে এবং উপকৃত হবে। বিশেষ করে যারা আজকে 6th সেম পাস করে বেরিয়ে গেছে, আশা করি তারাও যদি এই প্রোগ্রামটায় থেকে থাকেন তাহলে খুব ভালো হবে। আমি আশা করব ছাত্রছাত্রীদের কাছে এটা খুবই গুরুত্বপূর্ণ একটা আলোচনার বিষয় হবে, কেননা তারা এরপর মাস্টার্সে যাবে, সেখানে গিয়ে এগুলো করতে করতে এটা জানতে, শিখতে, বুঝতে, তাড়াতাড়ি বুঝতে পারলে তাহলে তাদের গবেষণার ক্ষেত্রে অনেক বেশি উপকার হবে। আমি আজকে আমন্ত্রিত দুই স্পিকার, ডক্টর রাজীব বন্দ্যোপাধ্যায় বর্ধমান বিশ্ববিদ্যালয়ের এবং ডক্টর সায়ক গাঙ্গুলি সেন্ট জেভিয়ার্স কলেজ কলকাতা, তাদেরও ভীষণভাবে কর্ডিয়ালি তাদের আমি আমন্ত্রণ জানাচ্ছি আজকের প্রোগ্রামে। তাদের ভ্যালুয়েবল বা মূল্যবান সময় আমাদের দেওয়ার জন্য। এবং আমি ছাত্রছাত্রীদের অনুরোধ করব, তোমরা অত্যন্ত গুরুত্ব সহকারে এই প্রোগ্রামটা দেখো। আমরা চেষ্টা করছি এই সময়ে অনলাইন ক্লাস তো আছেই কলেজে, তারা যারা রয়েছে তাদেরও আরো বেশি করে কিছু ইনপুট যাতে তোমাদের দেওয়া যেতে পারে, যেটা ভবিষ্যতে তোমাদের উপকার করবে সেইটা। কারণ এই আজকের বিষয়টা আমার সাবজেক্ট আলাদা, বোটানি নয় বা বায়োসায়েন্স নয়, আমি কমার্সের লোক, কিন্তু কোথাও আমি যেন এই বিষয়টার একটা সিমিলি খুঁজে পেলাম। হয়তো আমাদের হিউম্যান রিসোর্স ম্যানেজমেন্ট বলে একটা সাবজেক্ট আছে, যেখানে বহু দিন ধরে সংরক্ষিত হয়েছে একটা আকর বলা যেতে পারে, অর্থাৎ হিউম্যান বিহেভিয়ার। সেই হিউম্যান বিহেভিয়ার কোন সিচুয়েশনে কেমন বিহেভ করে, সেইটা যদি আমরা জানতে পারি, জেনে নিতে পারি, তাহলে তাদের ম্যানেজ করা অনেক সহজ হয়। এটাই হিউম্যান রিসোর্স ম্যানেজমেন্টের প্রথম কথা বা মোদ্দা কথা। তাহলে সেই বিভিন্ন সিচুয়েশনে, বিভিন্ন কন্ডিশনে নানান রকম কেস স্টাডি করে দেখা গেছে একটা ন্যাচারাল হিউম্যান বিং কেমন বিহেভ করে, এবং সেই রকম সিচুয়েশনে অন্যান্যদের তাদের যে বিহেভিয়ার সেই অনুযায়ী যদি ম্যানেজ করা যায়, তাহলে হয়তো আমরা ফ্রুটফুলি ম্যানেজ করতে পারবো। ঠিক তেমনি এই বায়ো ইনফরমেটিক্স হয়তো বেশ কিছু তথ্য বা ডেটা এমন ভাবেই সংরক্ষিত করেছেন, যেখান থেকে যারা এই বিষয়গুলো বোঝেন, জানেন, তারা তার পারমুটেশন কম্বিনেশন করে নানান মেডিসিন বা বায়োলজিক্যাল বা বায়োমেডিসিন নানান রকম তৈরি করছেন। এবং এনাদের সাহায্যেই তো আমরা আজকে এই প্যান্ডেমিক সিচুয়েশনে যে এই করোনা ভাইরাসের যে টিকা বা এগুলো পেলাম, অ্যান্টিডোটস যেটা পেলাম, সে তাদের সাহায্যেই পেয়েছি। সুতরাং ছাত্রছাত্রীরা, তোমাদের আবার রিকোয়েস্ট করবো, এরকম একটা সুন্দর অনুষ্ঠানে, মনোজ্ঞ অনুষ্ঠানে উপস্থিত হয়ে সম্পূর্ণ বিষয়টা জানার চেষ্টা করো। এবং সেই সঙ্গে সমস্ত টিচার এবং অধ্যক্ষকে আবার ধন্যবাদ জানাই আমার তরফ থেকে। ধন্যবাদ।
Thank you, sir. Thank you for such inspiring words. Now, I will request all the viewers to post their questions in the YouTube live chat box. Please mention the name of the resource person to whom you are addressing the question. The question answer session will be conducted after the completion of both the sessions, both the lecture sessions. Okay, now, as we are proceeding in our popular talk, I would like to briefly introduce you to our first speaker, Professor RAJIB Bandopadhyay. Professor RAJIB Bandopadhyay is a professor in UGC Center of Advanced Studies, Department of Botany, the University of Burdwan. Besides obtaining his doctoral degree, he holds postdoctoral research experience of 4 years in 3 different labs. The first one is to be the prestigious lab mentored by renowned geneticist Dr. P.K. Gupta and Professor H.S. Balyan of Chaudhry Charan Singh University, Meerut. The second one with Dr. Yoon in Jeonju, South Korea, and then from the University of Georgia, Athens, with Professor Andrew Patterson, till he joined the University of Burdwan. Besides, he is a life member of 12 prestigious National Academy of Sciences, Allahabad. He is the chairperson, lead speaker, associate editor, and advisor of Jharkhand National Academy of Sciences. He is also the executive committee member, vice president of AMI unit Burdwan. He is the recipient of several prestigious fellowships from just like DBT postdoctoral fellowship, Boy Scouts fellowship, fellowship from Korean Research Fund for Foreign Scientist Exchange Program. He has also participated in the Southern Ocean Expedition. He is also the state coordinator of International Science Congress, Jharkhand. Moreover, he has a teaching experience of more than 17 years and is well renowned among his students for his very dynamic nature. His research areas include characterization of different molecular markers from cereals as well as medicinal plants, Apart from this, he has also done molecular characterization of novel hydrogen-producing algae, and at present he is working on analyzing the extremophilic bacteria and is known widely for his experience and expertise in dealing with bioinformatics. So without further delay, I would like to invite Dr. Bandhopadhyay to deliver his talk. Please, sir.
Thank you, Dr. Seli. First of all, I want to thank Principal Dr. Prashanta Bhattacharya and IQAC Coordinator Dr. Tanmay Bandhopadhyay. And also I want to thank Head of the Department of Botany, Dr. Jaita Shaha, and Dr. Shumanti Guha. So we have with us with another speaker, Shayab Ganguli. He is a young and energetic person, though we do not meet face to face. But I hope Shayab work with Dr. Abhijit Datta. Is it so? Okay. So I have a hard name from Abhijit Datta because Abhijit Datta is my lab senior. And for several times when he was a coordinator at Presidency College for Bioinformatics Center, he visited our laboratory at PIT Mesra Ranchi. So from Abhijit Datta, I came to know SAYAK. Anyhow, actually today I am sandwiched with this program. I was attending one international seminar at LPU Punjab, and I was evaluating oral, and meantime I have to left because Dr. Seli told me Principal Sir is here, you have to join by 2:30. So another means things are there. My student, she has PhD defend at 3:30. So my talk will be very short. So I will request SAYAK, you can discuss with the students. You will get my time also. So you discuss freely with the students. And if our pre-PhD seminar is over by 4:30, means it may be 1 hour minimum, 3:30 to 4:30, I will again join. But if it is delayed, I cannot join. So Sayab, please you take care of this program so that our students, they should not lose the essence of today's discussion. Okay. So without wasting my time, I am sharing my slides. I cannot see the students here, so that is the minus point. I think they may be in the YouTube, and I don't know how many students they are attending it. So how I came in this field? Actually, when I was a student, PhD student in Calcutta University, under Professor Rajat Kanti Chaudhary, we used to work on plant molecular biology. I used to work on Ricinus communis, its gene sequencing, and many other protein or other biochemical marker, different aspects. So at that time, we do not have the access of email also. So for email, we have to depend on Calcutta University server, and we have to visit Rajabazar campus to access email. Otherwise, cyber cafe were there, the number were very less, and it was very costly, say 60 rupees or 100 rupees per hour or like that. I cannot remember exactly the things. So only one computer was there in the room of our HOD, Botany in CU. So in the working days, we cannot access that computer, and honestly, nobody accessed that computer during the office days. Only my professor, he is little bit friendly and habituated with the computers. Even in our personal lab also, we had a very old computers. I used to type over that computer. Even my professor had a typewriter. I used to type in the typewriter also. But Saturday, when the office is closed, my professor used to access that HOD's computer and the internet system. So Saturday was the amazing day for me because I used to come in the laboratory very first and I used to set up my experiments, and I used to wait when my professor will leave the computer. I will do something on the computer because it has the internet. So like this way, we learned better. I learned how to use bioinformatics and what is bioinformatics, what is database, what is NCBI side. And as I was working on the MadVox gene, so we have to submit the gene sequences to the NCBI, and I have to blast the sequences to see the similarities, dissimilarities, blah, blah, blah. So that is the beginning we did with so many difficulties. Then when I finished my PhD work, I joined with Professor P.K. Gupta. In P.K. Gupta's lab also during those time, means 2002, 2003, 2004, 5, like that time. He had internet system in the laboratory dedicated to structures. We have the work, we used to go to the cyber cafe. Normally we used to come in the laboratory, we used to set up the experiment, and it was instructed to us, those who have the bioinformatics work, you straightforward go to the cyber cafe, and we have the monthly system with that cafe. There are a lot of students in the laboratory. We used to use the facilities, and normally we used to pay at the end of the month. So that, like that way, we developed our bioinformatics. We worked there on wheat genomics, genome characterization, the repetitive DNA, the gene-rich regions, and making of different libraries, etc., etc. So like this way, I visited many places, many areas. When I was in USA, we worked on sorghum whole genome sequence. So anyhow, we need some bioinformatics. And like this way, whatever we are doing the wet lab research, we have to mix with some dry lab research also, or vice versa. So that was in my mind. And when I joined as a faculty at IIT Mesra Ranchi, we have the DIC, sub-DIC center, that is Bioinformatics Laboratory, funded by DBT, Department of Biotechnology, Government of India. And we have the facilities over there, so many computers, so many softwares, and paid softwares, and many, many things, and very good internet system with lease life. When I came to Burdwan, the position is means totally different. We don't have computers. I have my personal laptop, but department don't provide us any computers or internet facilities. Is very, very slow. And whenever summer is there or rainy season is there, the service is down till date also. Even if we have some important meetings or important things, we used to prefer to stay at home because we have Geo system, we have internet through Airtel also, but in the campus we have very, very slow signal. So anyhow, gradually, Government of India, that is UGC, they introduced some topic of bioinformatics at the UG level. This is very interesting, and this is obviously it was needed. So some of us in India and in abroad also, a lot of members, live members, advisors are there. We used to continue one platform that is called BioClues. BioClues platform we used to use to promote bioinformatics, and we even reach up to the school level also to teach students bioinformatics. And school children, they are performing good projects and they are participating in seminar with us, and they are competing with us, and they are getting good award also. At international level also they are participating. So my idea is that we have to introduce bioinformatics. So when I came at Burdwan University in 2014 October and 2015 onwards, I used to introduce bioinformatics slowly in the syllabus at PG level, not at UG level. So in the very beginning, with 1 or 2 years, it was really difficult for me to make our teacher make it understand what is bioinformatics, what is the need of this subject, how we can help our research like that. When I joined in the department, I used to see if some of our research scholars want to do a phylogenetic tree, they used to travel here and there to make that thing. So anyhow, 16 onwards, I used to teach bioinformatics and I used to make practicals for them. So now all my research, all my PG, MSc students, they know how to make the phylogenetic tree, how to do the analysis, what is protein docking, that little bit of things. So when I first started this thing, some of the students, he went back to home and he pressurized his father. Our new sir has come. He has told we have to purchase computer. Otherwise, he is not. He will not allow me in the class. So it was like that. So some of my colleagues asked me, "Have you instructed like this?" I don't know. I have not instructed like this. But that student, he made a false comment and he did like that so that his father will purchase a computer for him. So this is my personal experience. And even for last few years, and when it is pandemic, when it is means offline classes in the campus, I used to call the student. Most of the students, they don't have any computers. So in the department, we have very old computers and internet is not there like that. So I used to teach bioinformatics with the help of my research scholar. Mostly the research scholar, they have their laptops. So including my laptop also, there are say 6, 7, 8 laptops. I used to divide the student in group and they used to make when it is pandemic, say for last 2 years, so how I will teach bioinformatics to the students? So normally they used to depend on the smartphone. So I thought discussing with Sally, a lot of teachers in the colleges, a lot of faculties in the colleges, they used to make principle to understand that without computer we cannot do all these new subjects. We cannot run this subject at all without the computer. So it is the negative message to a lot of colleges. So I appreciate Rabindra Mahavidyalaya. They have keen interest and they are continuing so many popular talk and various type of interactions with different people. So it is very nice thing. So when UGC started the courses, so I may remember very few colleges. I think maybe 3 or 4 colleges just contacted me, said, "Can you start bioinformatics in our college?" I say, "Okay, you can start. If you feel any problem, you just let me know." Even Horipal Vivekananda College, I think in Horipal Vivekananda College is there, Sangram Sinha is there. We started the program, but neither he has the expertise or they have the computers. So I requested principal, you approach to our HOD through proper channel, and we will arrange their practical classes in our campus. Theory you can manage, and faculty they can manage, or we can do some workshop or program also for the faculty. So like this way, we are going to popularize bioinformatics in Burdwan University. So Calcutta University, Kalani University, and Vidyasagar University, all they have bioinformatics center, minimum BIF, bioinformatics facilities. But unfortunately, Burdwan University is so old university, and lot of faculties are there, different academic departments are there. Till now, there is no promotion of bioinformatics, unfortunately. Anyhow, so for the students and the young faculties, and my kind request to the principals are also, we can start with our smartphone. So nowadays, students, they have their smartphones for these classes. I am not going into the debate whether all students have smartphone or not. There are so many debate and so many data to me, mostly problem with online teaching in school level or at school level in the village side. There are problems. Anyhow, if we think the college students are a little bit matured enough and they have their smartphone, I think through smartphone we can start, and gradually when we'll get the computer, we will shift to computer. But if we avoid this subject, I think we will be illiterate in the coming future. Suppose when all other states, they are introducing computers, but the then government, West Bengal government, they thought if we introduce computer, people will lose the job. But the scenario is reverse. If you create the facilities, more people will get the job. You can see the IT sector in shortage. So we are late. When I was a MSc student, when I visit other places like Hyderabad and other places, I can see they are very computer friendly at that time, and automatic vending machine and location of different trains, these, that, all are available in their railway station. But in West Bengal, we don't have all those things at all at that time. So anyhow, like that, bioinformatics, we have to start with our small things. So these slides is for the students only. So bioinformatics, we will get the definition from different books. There are a lot of definitions, different things. So those who are newcomer, for them it is like that. It's the information, and this information is related with our biological things. So any informations if we have and related with the biological things, we can perform bioinformatics. We can do the analysis. And actually, it is the amalgamation of different subjects like mathematics, statistics, computer science, biological science, information science. So Dr. Seli elaborated nicely. So now the time of the big data or the data science, so people are getting degree, PG degree in say data science, or people going to abroad to learn how to handle the big data, because all these type of subjects are not available in India, mostly in the government system. Though some private system, they have the ideas. Bioinformatics is a major new stream in which application of high performance of computing, and you can see the applications of this bioinformatics is tremendous in the biology. Suppose in drug designing, when we talk about the wet lab experiment, normally the pharmacists, they used to say earlier, we used to take 30 to 40 years to release medicine into the market. But nowadays, within say 10 or 12 years, you can get that thing. You see the pandemic things, and people, how nicely within a very short period, they discovered these vaccines, and all we are getting that benefit. So bioinformatics, there are many synonyms like computational biology, computational molecular biology, or bioinformatics, or computational genomics, or biocomputing. It depends upon the person who is using it, who is using this thing. So before coming to these slides, I will give you another example. Say Professor GPS Raghava is a very big person in bioinformatics in India. So he's a Bhatnagar awardee scientist. You can call Nobel Prize of India. So I have several talks with Raghava. Actually, he told me that he did MTech. The same time he did MTech in computer science from IIT Delhi. So at that time, he's quite senior. So at that time, when he completed MTech in computer science, he got job at Intech Chandigarh. So he told, I used to sit idle. I don't know what is my work, what is my responsibility, what is my job. Scientists used to come, they used to say, you have to perform this thing in computer, you do this thing. And they are publishing the paper. So after seeing this thing, what they are doing, I can do that thing. So he started to publish papers. So like this way, he indulged into the field of the bioinformatics. So first, he did his PhD also, self-guided PhD in bioinformatics, data analysis, and gradually scientists used to give him challenge. Can you publish this paper in that journal? So suppose that has a big impact factor. So easily he can publish. So that is the thing. So right now he's a professor at IIIT Delhi, International Institute of Information Technology. So whatever the big project DBT used to give related with say any diseases or anything or construction of databases. Raghava used to do that job. So recently he has published one say software related with allergy. So for allergy, there is a dedicated database that is Allergom. So he did something else. So many people from different backgrounds, they started with a small, but nowadays you can think like Raghava, Bhatnagar awardee scientist and internationally famous worker in the field of computational biology or say bioinformatics. So we have plenty of examples like that. Some people they are from physics background, some people they are from chemistry background. They know good simulation and talking, this, that, and many things. But you have to start from a small thing so that you can reach to the that level. So in bioinformatics, we can do mobile learning or M-learning. So suppose nowadays the senior person who are faculty know about mobile banking also. So everything app that can be downloaded in your mobile and you can do the job. You can see all the government schemes, central government scheme or West Bengal government scheme, all they are doing is online. So everything you have to go to the URL, you have to click, you have to put your data, then you have to go for the registration, you have to upload your photographs or any kind of things. You may see admission in colleges and university also, everything you have to do through the computers or internet. Now, the era of internet, it is the era of internet. So you have to friendly with the smartphone or internet system and analyzing of the data. So we prefer say mobile learning or mobile things. Suppose if we want to sleep at night at 11 o'clock and I thought I have to check my mail whether there is important mail or not for tomorrow's work. So we don't open our laptop or we don't put on our computers because that is little bit time taking. But mobile, you just click one button and you see whether your mail is there or not, or you have any WhatsApp message or not, or if you have to reply something, you just give a quick reply and in details what later on you can do with your computer. So like this way, through mobile, we can collaborate with other people we can do the higher engagement, or it has the flexibility to the learners, or it has different performance support, and we can attach this mobile with different gadgets, with say laptops or desktop or anything. You can carry it easily while going traveling. You can work in your mobiles. So last say 2 years, we are teaching our students, and they are doing their projects and other work through mobiles, and they are doing their practicals also. So in biology, central dogma is there. You will read what is central dogma. We know DNA is our nucleotide, and from that we can get our protein, and this protein we will get the phenotype, different type of phenotypes. As we are student of botany, we can see the morphology, or any kind of anatomical data. So from gene to anatomy, from gene to morphology, we can correlate the data. So this type of information on, and the protein synthesis is very important. So in biology, these are the basic things. But first you have to understand. If you understand this basic biology, then you can design with your experiments. So nucleic acid sequence, when you will retrieve some sequence of some organism, or you will visualize their genome map. So with this DNA sequence, you can convert to protein sequence, or you can search the genes are there or not. You can check the promoter regions. Many more things you can design, you can check. You can count the ATG sheet. You can check the GC content of a sequence. Everything you can do. So RNA sequence, if you get RNA sequence, you can predict the RNA structure. So like this way, you can do many things. When you will get the protein sequences, you can go, you can do the reverse genetics also. From protein sequence to DNA sequence, like that. You can do the protein structure prediction, protein modeling. You can check the interaction, DNA-DNA interaction, DNA-protein interaction, protein-protein interaction, like that. So these are the typical protein. You can download this protein from the PDV site, that is protein data bank. There are many data bank like Swiss prot. We can classify database based on their structure or function, or any kind of things what is deposited over there. So like this way, what Sally was telling, we can go up to the drug designing. We can see their motifs, their protein-protein interaction, or many other things we can see. We can see the metabolic pathway in KEGG site, and we can go for the drug designing also. So FASTA is a very common term. It is called the first alignment tool. We can use the FASTA things. You can see here, this is the typical DNA sequences. Before that sequences, greater than sign will be there. There will be some unique numbers, and brief description of the sample from where it is taken, what kind of sequence it is a coding sequence or not. Like this way, we can see the FASTA part. So now I am showing you this screenshot. All are made in a smartphone. So when our student used to do the work, I used to say that you just click or you take the snapshot of the site, so that you can remember all these things. You need not to worry about the internet system or not. Suppose this is one side, this database, that is NCBI, it is in USA. So it looks like that. This is the home page. So if you have interest, you just click on different buttons and you see what is happening. So like this way, through self-learning, you can learn bioinformatics up to say 80%. So all we are actually bioinformatics users. So those who are hardcore bioinformatician, actually they have to know the programming. So you have to know C language, you have to know C++ language, or Java, or Bioform, like that, or Python. So those who knows the programming, they can develop the software, they can make it available for us, and we are the user, we can use that, but we don't know what is happening inside. So those who have keen interest with computer science, computer language, or mathematics, they can go into depth, and they can be the hardcore bioinformatician. But otherwise, like me, people like me, we can use the facilities of the software. So like this way, we have these PDB databases, we have EMBL, EBI databases like this. I have written the link also. So all these databases you can open in your smartphone. So like this way, next when we assign the work to our students, so suppose you open the NCBI site, you search for the nucleotide in your desired sequence, you can put the name, and then you can get the sequence in FASTA format. If you click a particular sequence, you will get this kind of whole sequence. You can download these informations and save the sequence in your word book or in your notepad, anywhere for further analysis. So when we will get the sequence, we can go for the pairwise sequencing, or we can go for the sequencing. So when we go for pairwise sequencing, the first step you can see, you have to go to the nucleotide things. Then you have to put your second nucleotide here. If you submit, you will get your done, job is done. You can see the result like this, because this is the pairwise alignment. So you can check manually, or you can check whether there are some polymorphism or not. Like this way, you can go through. So this is multiple sequences. When you have more than 2 sequences, you can overlap them. You can see their structure, their pattern, and if there is any mismatch, or if there is any gap or any penalty, you can show all these things in your smartphone. So this is multiple sequence analysis. You can go for the multiple sequences. Suppose we have chosen here, say 5 sequences. When we used to teach our PG student, we ask them to choose, suppose you are for taxonomy, you choose multiple sequence from plant. Somebody are from algae, you choose sequence from algae. Somebody are microbiology, you choose sequence from microbiology. Like that way, they can do this thing. Then we can go for homology modeling and phylogenetic analysis. So these are the slides where you can see how we can do these things. So then GC calculator. So you can see this slide, oligo calculator is there. So if you have a sequence, full sequence of DNA sequence, you can calculate the GC content, you can calculate the melting point, all these things you can calculate. Even you can go for their codon, you can see. You can see their molecular weight. So many more things you can see. So again, for the protein, you can go for the primary structure prediction and their physical chemical parameters for a given protein, their binding energy, all these things you can calculate. You can calculate their secondary structure also. We used to use, say, one thing that is called SOMA. So you can use this SOMA and you can get your result and you can see your secondary structure. So during this COVID time, post-COVID or pandemic time, mobile phones have emerged to be quite apt for teaching learning processes than laptop and desktop. a lot of our teachers, they used to teach through their smartphone, and even the students, they are also taking their classes through this smartphone. So as the site is small, so this is affordable and you can carry and you can use this thing, and it has some multimedia application. You can enjoy in a playful environment. Smartphone does prove to be readily available, and it is relevant to complementary tool for teaching bioinformatics. The approach has resulted in an empowering and inclusive experience and challenged the differences in the socio background and technological accessibility. So this is my last slide, and I will leave by 3 minutes. So this is the overall message I can give to our beloved young students, because you may be in the 6th semester or you may be at 3rd, 4th, any semesters. So in syllabus you have your means bioinformatics paper. You can choose this thing. Don't worry and don't fear. So anything new, if you can learn, no doubt your demand will be there in the market, and you can do many things. So I will stop here. I will not say much today, and we'll discuss later on. And if you need, I can send this slide to Sally Madam also, so you can read slowly. You can see different things and how it looks like, and you can do this thing. So thank you everybody for your kind patience and listening. Dr. Sally, I have just 1 minute. I have to leave.
Okay, sir.
So thank you,
sir.
Thank you, Principal Sir, again for giving me a chance to talk with your faculties and students.
Thank you, sir. Thank you very much for your lucid, student-friendly, and highly informative talk. Now, I would request my colleague and joint convener, Dr. Joyita Shaha, to please introduce our second distinguished speaker, Dr. SAYAK GANGULI, Assistant Professor, St. Xavier's College, Kolkata. Thank you, sir.
So taking permission, I am leaving everybody, and I will further request Dr. SAYAK GANGULI, please you continue and discuss in detail so that the student can enjoy, because I was in hurry and they cannot pick up everything. Okay, thank you everybody. Thank
I'll try my best. Thank you.
Thank you.
Joyita?
Yes. Am I audible?
Over to Joyita. Yeah, you are audible.
So thank you very much, Dr. Sinha, for giving me the opportunity to introduce Dr. SAYAK GANGULI. At present, Dr. GANGULI is serving as the Assistant Professor at the Postgraduate Department of Biotechnology of St. Xavier's College, Kolkata. He obtained his MSc in Botany with specialization in plant tissue culture from University of Calcutta and his PhD degree from West Bengal State University. He was the recipient of DBT Fellowship for Biology Teaching through Bioinformatics Scheme, Government of India at Presidency University. He also served as scientist of Amplified Institute of Interdisciplinary Science and Technology, Kolkata. Besides, he has experience of teaching and consultancy for almost 15 years. He is also the life member of different biological associations. He is acting as supervisor and co-supervisor of several ongoing projects funded by ACRB, ICMR, DBT, and WBDST. His scientific contributions include several international research papers, book chapters, and database submissions, invited talks in international and national levels. His research interest consists of areas like comparative and environmental genomics, tribal health, and gut microbiome structural bioinformatics, and drug design, development of data repositories, etc. So now I request Dr. Ganguly to deliver his talk, as we all are eagerly waiting to hear from him. Dr. Ganguly, please, over to you.
Thank you, Dr. Shah. And primarily, I would like to express my gratitude to all the conveners and joint conveners for inviting me, for sharing a bit of my work, as well as providing students with insights on bioinformatics. So let me share the slides first, and then we can Is the slide visible?
Yes, sir. It is visible.
Okay, thank you. Okay, so since we are here to talk about bioinformatics, I love to treat bioinformatics as an independent subject, because if we are not treating bioinformatics as an independent subject, then somewhere we are sort of transgressing the lines between the conglomerative potential of this particular discipline, since anybody who is associated with the natural sciences, be it from a mathematics background, be it from a physics background, chemistry background, biological science background, any biological science background, statistics background, can actually contribute and work in the field of bioinformatics. And just like every subject has a history, and bioinformatics itself has its own history and past, which unfortunately many of us are not aware of. So initially, I will try to take you down memory lane in terms of the history that bioinformatics has to offer. And any subject, if we are talking about the history of the subject, requires its coinage. And bioinformatics owes its coinage to a Dutch scientist, and her name is Pauline Hogeweg. Her group was the first to formally define the term bioinformatics. And in her seminal paper, which came out in 1970, she defined bioinformatics as the study of informatic processes in biotic systems. And how did they actually come up with this particular term? So Pauline Hogeweg was essentially an enzyme chemist, and she had accumulated a lot of kinetic data, enzyme kinetic data in her lab. And she needed something or some kind of machine learning process to evaluate and analyze the data, because the data volume was so huge that it was taking a lot of time to analyze that. So she approached her colleague, Ben Hesper, who had nothing to do with biology, who had nothing to do with chemistry. He was the early stage computer scientist. And Hesper developed a very short program in Fortran. Fortran is still one of the primitive languages which has then evolved into subsequent programming languages. So the first program was written in Fortran, which enabled Hogeweg to analyze her data. And this success led her to realize that indeed there was some element of computer that could be used to analyze biological data. And thus, she coined the term bioinformatics. So that is one of a very important piece of history that we need to know when we are embarking in our journey in the field of bioinformatics. Moving on, we then look into another stalwart in the field of bioinformatics, and she was Margaret Dayhoff. And she is credited to have developed the first manual database. Professor Bandopadhyay was talking about online databases. Think about an individual who has developed a manual database, and that manual database was credited to her and is still known as Dayhoff's Atlas. And this Dayhoff's Atlas consisted of the 65 protein sequences that had been sequenced at that time when she was developing this. So all these multiverse of contributions in the field of bioinformatics gradually led us to establish this particular subject as an important addition to the field of data analysis in biology. And that was initially what it was. It was a mode of computer applications through which you can analyze biological data. So that was the initial stepping points in the history of bioinformatics. And then we had the development of BLAST, the basic local alignment search tool, one of the most used computational tools that almost every biologist who has worked with molecular data has at least used once in his or her research career or academic career. So this basic local alignment search tool enabled us to compare, which forms the fundamental of biological research. We compare between phenotypes, we compare between processes, we compare between gene expressions, we compare between sequences. So this was that kind of a tool which enabled us to compare between sequences. And this has been one of the citation classics in terms of the journal publications, and currently has more than 4,000 citations for this particular paper. So you can understand that the significance of BLAST in terms of the biological data analysis. And if we take a look at the Bioinformatics Hall of Fame, so this is a collection of individuals who have contributed richly to the field of bioinformatics. So we have already talked about Pauline Hodgweg. We have already talked about Margaret Oakley Dayhoff. We have already talked about Ben Hesper. Who are the other three? I talk about Robert S. Ledley first because Robert S. Ledley discovered or developed something which has been hugely discussed in the media for the past 1.5 years since the pandemic struck us. And he was the first to develop the concept of CT scanning. And he also formulated a book in which he described the process and also accounted for the application of computers in biology and medicine. So that book came out in the year 1965 or 67 and has been one of the richest sources of computer applications in medical sciences. The other two, one who should be known to us all because he is an Indian, G.N. Ramachandran, and people who have worked, or if you have studied protein structures, then you would have come across a concept called Ramachandran plot, which enables us to get an insight into the conformational stability of a particular protein. And G.N. Ramachandran, in his work, established the phi and psi angles, which forms the basis of the Ramachandran plot. Finally, we come to Frederick Sanger. And again, a basic biology student having an idea regarding the concept of DNA sequencing is well familiar with Frederick Sanger. Frederick Sanger established the first process of DNA sequencing through his chain termination process. And he is regarded as the pioneer of DNA sequencing technologies. Remember, we will talk about DNA sequencing technologies and their evolution in the later half of this particular discussion. But Frederick Sanger needs to be remembered as one of the pioneers. And it was his method of DNA sequencing, which was then further automated, which was used for the generation of the sequence of the human genome. And the Human Genome Project owes its success to Frederick Sanger. So if we are really interested in knowing regarding the history of bioinformatics, then in every history, you will find that these are the people who are mentioned for their contributions towards the development of the subject. So this is where we start our discussions in terms of bioinformatics as a subject and how it has progressed over the years, expanding its horizons. So we come to the coevolution. And this I term as coevolution because in biology, we talk about coevolution, we learn about coevolution. People from zoology might be learning about coevolution when they are studying the Red Queen hypothesis. People from plant sciences, botany, might be learning about coevolution when they are studying plant pathology, where they are learning about the gene for gene hypothesis. So biology has a rich history of coevolution, and so has bioinformatics. As you can see from this graphic, we are talking about the development of the different methodologies and a significant step in the formation of the bioinformatics pipelines. So we start off with the Watson and Crick DNA model. Then we have the sequence of insulin protein, which was developed and sequenced by Sanger. Then we have the development of the Dayhoff's Atlas, which I was talking about a few slides back. Following that, we have the ARPANET, which was developed in US within a particular laboratory. So this was the first evidence that yes, 5 or 6 computers can be connected together to share a network, which later was developed into the World Wide Web, which we now use today in terms of internet throughout the international community. So that is the first development of ARPANET. We come across the sequence alignment, the concept of sequence alignment, and we have algorithms for pairwise sequence alignment. We have algorithms for multiple sequence alignment. We have local alignments. We have global alignments. We have Needleman-Wunsch algorithm. We have Smith-Waterman algorithms. So all these algorithms, methodologies by which we could compare comes under the purview of your sequence alignment. Then people started realizing that yes, we are in a position to develop and generate data. And we require essential systems, repositories, dedicated addresses where we could store data. And as this realization dawned upon us, we have the development of databases. We have the development of Protein Data Bank, which is the collection of crystallographic and structural data pertaining to proteins, DNA, RNA, etc. So a dedicated database which would serve as a one-stop repository for people who are working in the field of structural biology. So Protein Data Bank became one such repository, and it was established around 1975. Then Sanger sequencing established itself in 1977. And as we were able to sequence DNA more effectively using Sanger's method, we could develop the Genbank database. This database currently is housed under NCBI as a data warehouse. But this GenBank database at that point of time served as a collection of nucleotide sequences. It still does. It is one of the richest collection of nucleotide sequences that have been generated using experimental methods in the wet lab. So the GenBank database became formulated. Then you are all aware, people who are from the 6th semester probably are all aware regarding the PCR or polymerase chain reaction and the Nobel Prize winning discovery by Kary Mullis. And that almost went hand in hand with the development of such databases, enabling us to formulate the formation of the SwissProt database, which at the onset was a collection of protein or amino acid sequences. So within the year 1985, 80, 88, we had a collection of structured databases. We had a collection of nucleotide sequence databases, and we have a collection of amino acid sequence database. So our reference databases were ready, and we were ready to plunge into the field of bioinformatics. And that was initiated by the Human Genome Initiative. And this Human Genome Initiative paved the way for development of novel bioinformatics tools, development of novel methods by which we could store the huge amount of data that would be generated using the Human Genome Project. And hand in hand, you could understand that we could develop the European Bioinformatics Initiative Archive, or the EBI-EMBL archive, which still exists. We have the development of the BLAST, as I was talking about earlier as well, as an important tool which enabled us to compare all the sequences that were being generated through the Human Genome Project. Finally, we had the establishment of the World Wide Web. And this World Wide Web enabled us to exchange data. And this was one of the very essential developments towards the successful completion of your Human Genome Project. Because as you all know, the Human Genome Project was being contributed by several different laboratories spread all across the world. Together, they were generating data which would be compiled together and formulated into our human genome or reference genome. Along with that, we had the development of your first bacterial genome, the development of the yeast genome, the development of the first human genome draft, which was published in the year 2002-2003. So you could understand that as the nature of biological data has progressed, has changed, has evolved, has developed into humongous proportions, so has the need for computational bioinformatics-based algorithms, data structures, systems for storage of data, having contributed equally towards such initiatives. So both have progressed hand in hand. And if these wouldn't have progressed hand in hand, then the success stories that we now read about in terms of these several genome projects wouldn't have been possible. So that is one of the key facets which we need to remember when we are talking about bioinformatics. And this was very briefly talked about by Professor Bandopadhyay. And I will just compare how I perceive bioinformatics as. So we are in our quest of understanding the genotype to explain the phenotype, and the other way around. So in our journey from genotype to phenotype, we have the DNA or the genome, which codes for the RNA, the transcriptome, the protein, the proteome, the molecular network, the metabolome, the cells, which contribute, is a collective function of the molecular networks, and any aberration results in a disease condition. So we are taking into account all these facets when we are taking into account the data sources that we talk about when we are embarking upon a bioinformatics analysis. Hand in hand, we have the data and the discovery. So we have the informatics angle to it as well, not only the biological angle. We have the informatics angle, which talks about the data management, how we are going to organize this data. Suppose you have a huge amount of syllabus which you have to cover in your exams, and each syllabus is divided into several papers. So when your professors are teaching you, you take down notes and then you arrange them either in the copies or you arrange them in files. So that is nothing but data management. You are managing data that you are getting. Similarly, here also we are managing data. So one of the fundamental aspects of bioinformatics is how you are going to manage data. The next aspect is data computation, which requires knowledge of both biology as well as computer science, or basics of computer science and very important aspects of biology. So biology and computer science knowledge is very important when we are going in for data computation. Then we have data mining. So what kind of data do you want for your experiment? Are you sure that this is the kind of data that you want? Are you absolutely fine with the kind of data that you are getting from a particular database? Or do you need to explore further? So that is one of the important questions that we need to ask when we talk about data mining. Finally, we come to modeling or simulation. Now, modeling and simulation as terms have been used in field of bioinformatics in various ways. One of the most common usage, and which we will talk about briefly later on, is when we are generating structures of protein. The other usage of modeling and simulation is whether you are actually able to model a disease process or a biological process using various variables. So that comes under the purview of systems biology, which is a foster child of bioinformatics or computational biology, which enables you to model ecological processes, model physiological processes, model disease phenotypes, and predict whether a particular disease phenotype can be prevalent based on certain issues or certain factors which you are considering for as a data input for your model. So both modeling and simulation is a sort of predictive framework which can be generated from the data that you are accumulating, which leads us to a sort of formal definition of bioinformatics. And this is sort of the definition that I would prefer bioinformaticians to assign to bioinformatics, where we are talking about the storing, managing, and logical interpretation of the vast amount of biological data with the help of information technology. And when I talk about biological data, sorry, I'm talking about sequences, nucleotide amino acid sequences, sequence associated data, ESTs, express sequence tags, STS, sequence tagged site, ISSRs, intersimple sequence repeats, which can be obtained by analyzing sequence data. Then we have structured data. We have 3D structures of proteins, crystallographic data in the forms of DNA and RNA helices, metabolic pathways, induced expression data, interaction data, pharmacological data, and ecological diversity data. And for students, remember that in the next 5 to 10 years, with the advent of the climate crisis, how computational biologists will work in analyzing ecological diversity data will become one of the key cornerstones towards the success and survival of humankind. This will be one of the most burgeoning fields in terms of computational analysis and computational data analytics. So climate change has actually enabled us to consider analyzing ecological diversity data, which leads us to 3 pertinent questions. And whenever we are talking about bioinformatics, we are talking about bioinformatics analysis, these are the three pertinent questions that you need to ask. So where do we get data? We are talking about data. All through this particular 10 or 11 slides, I've been talking about data. But where is this data available? How do I get the data? Do I have to generate data by yourself? That is one of the first questions that you need to ask. And we will see take a look at the answers in the subsequent slides. The second question is, can we design an experiment in bioinformatics if we do not have any self-generated experimental data? So this has been one of the prima facie questions that has been asked over the past 1.5 years more seriously, especially under the pandemic conditions, because experimental laboratories mostly have been closed. We have not been able to come out of our respective residences. Yet people who love to do research have been doing research. So how have they been doing research? They are not in a position to generate raw data by themselves. So they will have to rely on data that is already existing. So that becomes the second question, as if we are in a position to design an experiment, if we do not have our own data. Yes, bioinformatics allows you to do that. And third, Professor Bandopadhyay was talking about the two facets of a bioinformatician. One, where a bioinformatician is an user, and the other, where the bioinformatician is a developer. So for classical students of biology, you start your journey in bioinformatics as an user. When you are starting your journey in bioinformatics as a user, you gradually get to understand which particular aspect of bioinformatics you are more comfortable with. And then it is up to you to gradually develop your skills so that you can become a developer who can design algorithms, who can develop softwares, which will enable further users to analyze their data. But first, you have to get into the field as a general user. So that is how we approach a bioinformatics problem as a general biologist. You first, at the entry level, you are a user who needs to know which are the software, which are the servers, which are the resources which you need to use so that you can actually analyze the problem that you are facing, the biological problem that you are trying to solve. So these are the three pertinent questions that needs to be asked. So as a response to the first question, where do we get data? We get data in primary data repositories. And the four databases, the screenshots of the four databases that you can see here are the fundamental data repositories from where you can get a majority of the computational bioinformatics data that can be analyzed using computational biology. The first is NCBI, the National Center for Biotechnology Information, which is housed in Bethesda, USA. And they have a large collection of multiple databases. If this was not a talk and a workshop, I would have showed you the various different databases that NCBI contains. And hence, NCBI is often referred to as a data warehouse, since it is a collection of multiple databases. Then we have UniProt, which I talked about briefly when I was talking about the coevolution. The UniProt knowledge base has been developed or has superseded the SwissProt repository. And this is now the richest collection of amino acid sequences. On the bottom two columns, we have PDB and MIRBase. PDB is, as I said earlier, a largest collection of crystallographic data pertaining to proteins, DNA, RNA, small molecules, protein small molecule complexes, protein DNA complexes, RNA protein complexes, so on. Anything related to structure, you first go and search in PDB to check whether a crystallographic structure data is available in terms of the search string. Finally, we have MIRBase. So MIRBase is the richest collection of microRNA belonging to different organisms. MIRBase houses the collection of mature microRNAs as well as precursor RNAs. And it is one of the most trusted collections or databases where you can access, which you can access and come up with data pertaining to microRNAs. So that is one of the key aspects of these are the 4 primary repositories where we can actually see and search data. As I move along, there has to be a brief connection between the next set of databases that we will come across. And since these are primary data repositories, as I said, there has to be some specialized data repositories as well. And to form a bridge between the primary data repositories and the specialized data repositories, we have to take a brief look into our concept on big data. And this is something which has hit us hard, the concept of big data. That big data could be utilized or will be applied to biology was unknown to most of us, say around 10 years back. But big data has really hit biology hard. And when big data has hit biology hard, we have been able to identify 4 most important concepts of big data. The first being volume, the size of the data, which is huge. The velocity of the data, the speed of change. So you have a data pertaining to a particular experimental condition in day 1, in day 2, or in a week's time, you might find some additional data being added to the already existing data. So the speed of change of data has been humongous. Then we have veracity, the uncertainty of data. How data will vary. A particular experiment might give you a different kind of data. The same experiment conducted elsewhere is giving you some other kind of data. So which data would you rely upon? You have to think about these when you are analyzing big data. And finally, the methodologies for generating the big data has enabled us to understand and explore different sources from which data could be obtained. So the variety has increased many folds. And hence the 4 V's of big data, as I prefer to call them, volume, velocity, veracity, and variety, has actually forced the computational scientist or computational biologist to develop mature algorithms which would be able to analyze such big data. And the 2 instruments that you can see in this particular slide, one is the Minion and the other is the Smigion. So Professor Bandopadhyay was talking about bioinformatics data analysis using mobile phones. I am talking about DNA sequencing and analysis using mobile phones. And Smigion is a device which utilizes a particular technology which allows you to analyze samples using your smartphone. So Smigion as a device is not larger than a pen drive. And it can be, as you can see in the image here, it can be smartly connected to your smartphone using an USB port or a Type-C connector. And if you have the right kind of samples and you have the right kind of buffer systems or chemicals, you can prepare a particular sample and you can analyze or generate sequences using Smigion and analyze them using the accompanying software in your smartphone. So that And the Minion technology, which is the precursor of your Smidgen technology, allowed public health experts to intervene timely when the Ebola pandemic established itself in 2014. And it affected the African and a few coastal American subcontinents. It did not spread over to India, thankfully, but Minion enabled us, the public health scientists working there, to identify pockets of infection very fast and very quickly. Both these devices use a technology which is referred to as nanopore sequencing. And if you go and search in Google, you will find various informative videos in YouTube in terms of nanopore sequencing, which will explain why these devices allows you to generate data very quickly. And that is what has enabled data generation. So as I said, the 4 V's, you can understand, if you have a device which will enable you to sequence DNA in your pocket, then how much data you can generate in a day. So that has actually enabled us to generate a large volumes of data. I'm not going into the details of this particular section of this slide because that would make it more technical. And this deals with a classical NGS analysis where we are talking about the various file formats. I'll just put your attention to this particular segment, which says FASTQ file format. Professor Bandopadhyay talked about the FASTA file format, which is the classical file format for storage of sequence information. And if you access Genbank database, if you access SwissProt database, you will find that the amino acid sequences, the nucleotide sequences, which you can download from these databases for your personal usage or analysis, are stored in the FASTA file format. Interestingly, when you are dealing with a next generation sequencing analysis or next generation sequencing data, the data comes in FASTQ file format. So there is the slight difference between the FASTA file format and the FASTQ file format. I'm not going into the details because that is more of a technical lecture. So what have these techniques and technologies contributed to? They have contributed to biological big data databases, which are sort of secondary databases in terms of the kind of data that is housed in them. For example, you have probably you have all heard the term epigenetics. And here, just like the Human Genome Project, we currently have the Human Epigenome Project. And this Human Epigenome Project database provides you rich information in terms of the various facets of epigenetic control. So does your database on Encyclopedia of DNA Elements, or ENCODE, which gives you a very good idea regarding the regulatory elements that are there in our human genome. So that is one of the collections which you can explore if you are willing to work in the field of epigenetics. Then we have a very interesting collection called the Thousand Genomes. And as the name suggests, you understand that the repository was developed with the concept of achieving 1,000 complete genome sequences. Now it has more than 3,500 genome sequences that are there deposited in the repository. But it started off with the objective of contributing 1,000 genomes into that repository. And these genomes would be from different ethnicities all around the world. It would not be concentrated to a particular ethnicity, or for example, say Indian ethnicity or American ethnicity or African ethnicity, to make it to simplify it. And here you find genome information from human beings belonging to different ethnicities. So if you are really interested in working in the field of human population genetics, if you are trying to understand the progression of a disease across evolution, across human population, then this is one particular database which will give you good information in terms of genomes across the world. Simultaneously, we have the international Hapmap project, which gives you information regarding the haplotypes, the different population haplotypes that are there in terms of the information of single nucleotide polymorphisms, insertions, deletions. And the other three are related with microbiome research. The first is the Human Microbiome Project, which allows you to explore the composition of the microbes that are there in the different niches of our human system. For example, the gut, for example, the oral cavity, for example, the skin. All these have been explored, and it has been identified that as an individual, if you are, say, for example, if you are considering me as an individual, then my skin will have a different microbiome composition as compared to your skin. My gut will have a different microbiome composition to your gut. My oral cavity will have a different microbiome composition to your oral cavity. And just remember, take into account all the advertisements that come on television, which tell us to brush our teeth. And we keep brushing our teeth day in and day out, and yet there are microbes. So just think about it, the diversity and the kind of data that can be there when you are exploring different niches. The other two are the Earth Microbiome Project. So you are expanding a horizon further, where you are now moving away from only human system, you are moving towards Mother Earth, and you are exploring the different ecological niches of Mother Earth. So you are going to the Antarctics, you are going to the deserts, you are going to the mangrove forests, and you are collecting data, and you are analyzing the microbiome composition. Along with it, there is the Terra Genome project, which is specific to soil microbiome, and it deals with the various different kinds of soil associated microorganisms that you find all across the world. So these, and you can add the human pangenome database to this particular collection as well. I have not mentioned it here because it directly doesn't come under a particular big data database. But there, and if some of you are abreast with modern developments in science, very recently a news feed in Nature came out which told us that the gaps which were there in terms of the positions which could not be sequenced in the human genome via the Human Genome Project has been achieved by the T2T consortium, telomere to telomere consortium genome project. They have been able to fill up most of the gaps that still remain in our human genome. So we now have actually achieved the sequence of a complete human genome. So all these facets that I'm talking about are parts of your big data and the contributions of big data analysis in terms of data repositories and computational biology. So now you have more databases to compare your data with. If you are generating data via an experiment or a wet lab experiment, then you have much more repositories to compare your data with. So what are we, and now I'll just sort of show you some glimpses since the event is termed as Glimpses on Bioinformatics. So I'll show you some glimpses of data. So what kind of data we can achieve or what we can do when we are trying to analyze biological data using computational programs, computational tools. So what is the basic thing that a computer does when it is fed with biological data? Remember, we have letters to understand. A DNA sequencer has letters to understand. We have 26, they have 4. If you are sequencing an amino acid, then you currently have 22. So there are letters which we all understand, but a computer understands only 2 numbers. It's 0 and 1. So you have to make the computer understand whatever you are trying to make it understand using 0 and 1. So basically, what a computer does when you are feeding it with data, it is basically looking for patterns. And I came up with this analogy, and this was one of my favorite childhood books, and I believe most of you have read this, Sukumar Ray's Abol Tabol. And we had a very famous poem in that, Gof Churi. And in that poem, as I quote, Gof ki bolo tomar amar, gof ki karo ke na, gofer ami gofer tumi tai diye jaiche na, unquote. So what the poem told us could be compared with what a computer understands. The computer is only looking for patterns. And just like the pattern of your mustache will enable someone to identify you, similarly, biological data also houses several patterns through which you can actually segregate, analyze, and identify biological data. So basically, every computer program in any kind of bioinformatic analysis that we are talking about is searching for patterns. So if you keep this in mind, then this will enable you to understand how each and every data is being analyzed, each and every data is being generated, and each and every data is being compared. So ultimately, we are all searching for patterns. So this is an example of the secondary structure of an RNA. And as you can see, the best way to make out whether the sequence belongs to a DNA or an RNA is the presence of U, or the uracil. So this has been generated using an algorithm which is referred to as Nusinov's folding algorithm. As soon as I say the algorithm, people will try to run away. Oh my God, it is sounding very complex. But the tool that has been used to generate this structure is referred to as mFold. So this gives a more soothing ring to our ears. Just compare Nusinov's algorithm and mFold. Which sounds soothing? Obviously, Nusinov's algorithm won't sound soothing. mFold will sound soothing. So mFold is a tool which enables you to generate secondary structure of your RNA. So if you have an RNA molecule, then by just putting the RNA molecule into the sequence, into the server, you can generate a structure such as this. And I have just marked the components of the structures for your convenience. So typically, in an RNA molecule, in a secondary structure, you are looking at a stem or a stack-like structure, the ladder-like structure over here. You have an internal loop which connects the 2 hairpin loops which are projecting out. So this is how a particular secondary structure of an RNA looks like, and you can generate that using a server called mFold. You can also analyze this structure for its base pairing patterns. You can count the number of Watson-Crick base pairs that are making up the individual stem or loop-like structures. And you can also find the wobble base pairing pattern that is making up this structure. So this gives you a very important insight as to how you can generate the secondary structure of an mRNA or microRNA or any kind of RNA that you want to work with. So this is maybe one of the basic analysis that you can try out. And yes, you can do this in your mobile phone. And we have sort of compromised the efficacy of the data generation processes. And as a result, the next generation sequencing pipelines or next generation sequencing processes that are conducted in the wet lab produces a huge number of data, huge data, 3 GB, 4 GB, 10 GB data. So I'm talking about in terms of the gigabytes. And all these data is nothing but sequences. So what are you doing? You are generating DNA sequences. So you can understand from an organism, you are starting to generate 10 GB of DNA sequences. So if you are counting it manually 1, 2, 3, 4, it is near to 250 million sequences, which is contributing towards that data volume. Now, do you think that all these 250 million sequences are being sequenced correctly? It's almost near to impossible because there will be errors in the machine. And there is an essential step which is standardized across pipelines, which is known as quality control. And through this quality control step, we can actually eliminate certain sequences which are easily identifiable as being errors. So your quality control pipeline, after you have generated the set of sequences, allows you to exclude certain set of sequences which are erroneous. But there still remains a large collection of sequences which does not have any validated biological wet lab experimental evidence. And such sequences, if are found to be containing an open reading frame, I hope you are all aware regarding what an open reading frame is. So an open reading frame will code for a protein. That is our understanding regarding the identification of an open reading frame in a sequence. So this open reading frame will code for a protein. However, when we are doing this, how we are doing this? So we have generated the sequence, we have identified whether a set of sequences contain open reading frames in them, and we have been able to predict the protein that might be coded from that open reading frame. So now we have a protein sequence with us. However, we do not have experimental evidence that this protein exists. We have not been able to characterize this protein using wet lab experimental techniques. Hence, such proteins are referred to as hypothetical proteins. And if you go to NCBI, just type in Google NCBI, you will be directed to your NCBI database. And within the search box in NCBI, type in hypothetical proteins. And you will find that you are in a data deluge. Huge quantities of hypothetical protein data is available in NCBI. So why do you think it is available? Mostly it is available because such sequencing projects are going on all around the world. And whenever such sequencing projects are going on, you will find that such protein sequences are continuously getting updated in the database. So it is essential that we try to assign function to a hypothetical protein. But we are stuck in a pandemic. We are not able to access the essential wet lab facilities that would allow us to check whether that particular protein exists in that organism or not. So can we do it computationally? Let us take a look as if we can annotate. And here is the significance of the term annotating hypothetical protein. Annotation in computational or bioinformatics terms, annotation is saying that we are assigning function to a particular sequence. So we are annotating hypothetical proteins. So how do we do that? We do that using a very simple approach, and we use a set of tools and databases which allows us to explore the function of the hypothetical proteins. So you can see here in the middle column, we have two databases, CDD-BLAST and InterProScan. Both are tools connected to databases. So CDD-BLAST allows you to identify conserved domains in your sequence. So proteins, if you have studied protein structure, you must know that proteins have domains. So each domain is an independent folding unit. Now, this CDD-BLAST page allows you to identify conserved domains. Next, the interpro scan scans the entire sequence that you have submitted and comes up with information regarding conserved domain, as well as whether that conserved domain belongs to a particular evolutionarily conserved protein family or protein superfamily. So that is how you can do that, or that is a kind of data that is provided by interpro. What does the other servers allows us to do? One, we have the signal P server, which predicts the presence of signal peptides. Now you all know that once translation is over, protein folding and protein localization is initiated in the cell. So a particular protein might be synthesized in the cytoplasm, but it must be localized in the, say for example, chloroplast to be functional. So this transfer of that protein from the cytoplasm to the chloroplast requires the presence of a transit peptide, which is often referred to as a chloroplast transit peptide, and that is nothing but a signal peptide. So your signal P server allows you to identify whether your sequence has such kind of a signal peptide, which will enable you to understand the actual location of the function of that particular protein. Then we have Cello, which also tells you regarding the subcellular localization of the protein. So you are validating the result that you obtain from the signal P server. If you get a result, you might not get a result from the Cello server. And on the right hand side, as you can see, we have a server which allows us to explore protein interactions. So this protein interactions allows us to explore what kind of other proteins my protein of interest interacts with. That also gives me an idea regarding the identification or identity of that particular protein. And finally, we can compare it with an evolutionary ASP server, which is the eggnog server, which allows us to identify orthologs. And if we are able to identify orthologs, then we would have an idea regarding the function of that protein in some other organism. So that is the fundamental basis of your ortholog prediction. And if we get data from majority of the servers, we will be in a position to functionally annotate a particular protein. So let us take a look at some of the at an example in terms of this pipeline. So we have started off with a protein from Shigella flexneri. So Shigella, as we all know, is the pathogenic microorganism which causes dysentery. And we have started with a hypothetical protein from Shigella flexneri, and we have submitted it to the CDD conserved domain BLAST server. And it has given us a result which tells us that it is part of the PAR-B family. Now, PAR-B family is a set of proteins which is functional toward genome partitioning. So this is an information that we have already got from the CDD BLAST. So our first step towards the manual annotation of the protein is successful. Then we move ahead and we go to the interpro server. And as you can see here, the interpro server has given us large number of hits, which all can be accessed through these individual accession numbers, which you can see here, or links which you can see here, and will give you, if you access them, you will find that all of them is giving you information regarding the PAR-B superfamily only. So your CDD BLAST data and the data that you are getting from interpro is validated with one another. So you are able to justify the result that you are getting from one with the results that you are getting from the other. So tools of analysis. Then we are moving on to the signal P server. So in this signal P server, we find that there are no signal peptides that are predicted. And hence, this other segment here has a high score. Any signal peptide, if there was a signal peptide, we would have got high scores in either of these three segments. But we are getting a high score in case of this particular segment. Then we get to the cello results. So here you can see that it is predicting that it is a cytoplasmic protein. The localization of this protein is cytoplasmic. So that is one of the predictions from cello. Finally, we go to the strings database, which is a collection of the interactions, which gives us an information regarding the interactions of the individual protein. And here you can find that this is your protein which you have submitted, the central red circle that you can see here. And you find that it is giving you quite a number of interactions with other proteins as well, details of which you can obtain in the associated table that is there. So you can see that it is interacting with an acetyl transferase. You can see that it is interacting with a GMP synthase. You can see that it is interacting with a DNA polymerase. So all these interactions gives you more fillip to the initial data that you obtain through CDD, that it is a partition protein. And then you move on to the eggnog server. So as I said, we are looking for orthology predictions. And from the eggnog server, we get that it is orthologous to proteins which are present in Ophistochondra, Chrysotulus, mammals. So that's an interesting data. You started off with a bacterial protein, and here you are finding something which has similar function in mammals as well. So that gives you a very interesting insight as towards the function of the protein. And if you look closely towards the function, you find that it is involved in DNA binding. It is involved in polysome binding. So these functions enable you to understand that indeed the result that you got from your CDD blast might be correct. And so you move on to a phylogenetic tree, which Professor Bandopadhyay was also saying. And here you find, so this is my sequence of interest, which I started working with. I have renamed it as test sequence for you to understand. And here you can see that this test sequence is housed in different sister groups. So all these segments are referred to as sister groups. And here it is closely related to this particular protein of interest. And if you search with the accession number of this protein, you will find that it is a protein which belongs to Chrysotulus. And you can search in Google what Chrysotulus is. It will give you a very interesting insight. So you are at one end, you have started off with Shigella, and at the other end, you end up in Chrysotulus. Evolution is truly amazing. So finally, you come up with the formulation of a data matrix. And here you are incorporating the information that you are getting from all the tools that you are analyzing. So you have the test sequence. And from the test sequence, what information have you got from CDD blast? You have found that it is a PAR-B partition superfamily of proteins. From Interpro, you have found that it is a PAR-B, REP-B, SPOJ partition protein domain containing protein. From Cello, you have identified that it is localized in the cytoplasm. Next, in Signal P, you have not been able to identify anything. So fine enough. Move on to String. String, you have found that it is similar to PAR-B protein of Shigella flexneri and interacts with all the other proteins that have been identified in the network that I showed you. And in your eggnog prediction, you find that it has a closest orthologue from Chrysotulus. So this enables you to provide a final annotation towards your protein, that the protein belongs to PAR-B partition superfamily, having this kind of a domain, which is involved in interaction with other proteins and regulates cell cycle and cell division by binding to DNA sequences adjacent to origin of replication, thus controlling chromosomal partitioning. So now you are in a position to say that, yes, I have been able to provide a functional annotation to a sequence which was unannotated at the onset. And this will also allow you to design your wet lab experiments if you are trying to find a function of this kind of protein through wet lab analysis. So this has given you a complete idea regarding what that protein might be. And that is the power of computational or bioinformatics analysis. It gives you the opportunity to annotate, assign function to an unknown. So that is which we all fear, the fear of the unknown In biology, as biologists, we all fear the fear of the unknown. What kind of sequence it is? How are we going to design an experiment through which we are going to generate this kind of data? Because funds, as all researchers will tell you, that funds are in real shortage. Government agencies don't give us funds to work with. So we are always concerned about wastage of chemicals. So you have to think about it in those terms as well. Bioinformatics offers you that kind of an opportunity to validate your results first. To the organizers, do I have another 10 minutes?
Yes, sir. You can continue.
Okay, thank you. So next we move on, and we move on to the structure part. So what are the various things that we can do when we are dealing with structures? And one of the main important things that we do in case of structural bioinformatics is known as comparative modeling. Again, an unknown, we are working on an unknown. So we do not have, you have gone to PDB, and you have searched for the structure of a protein of interest. Say you have studied regarding DNA replication in your syllabus probably. So you have gone and searched for, say for example, gyrase, and you find that there are no structures available in gyrase. And yet you are interested to explore the structure of gyrase. Bioinformatics allows you to do that. And bioinformatics allows you to do that by employing the process of comparative modeling. And again, I have an analogy to our Durga Puja. And most of us probably have seen the gradual development of the Durga idols. So if you are trying to find analogy with comparative modeling, you find that you are gradually adding in layers of information, just like the artisan continuously adds in layers of materials to finally come up with the goddess. Here, what you do, we start off with a sequence. We search for suitable reference points through which we can compare. Do we have the information of another gyrase structure which might not belong to the organism from which I am interested to study it, but it maybe belongs to another organism? So can they be compared? So if they can be compared, then the structure with which you are comparing your sequence with is referred to as a template. So that makes your job easy. So you have a template with which you can compare. Information from the template can be taken to the sequence and you can generate a structure by yourself. But what happens if you do not have a template? Can we go ahead with that? Yes. That also is referred to as ab initio modeling. So you have comparative modeling and you have ab initio modeling. So comparative modeling, you have a reference template. Ab initio modeling, you definitely do not have a starting template. You are collecting information by analyzing homology with multiple templates, and you are taking in information into your sequence and you are generating a structure from your sequence. So comparative modeling obviously gives you better results as compared to ab initio modeling. And remember, I talked about Ramachandran plot. There, after you have generated the structure, how are you to evaluate whether the structure that you have generated is actually a proper structure? For that, we have to rely on G.N. Ramachandran and his Ramachandran plot. So there, you have to generate the Ramachandran plot using the structure that you have generated using either of the modeling processes. And your Ramachandran plot will give you information as to what is the number of outliers that are there. How many of the amino acids that are there in your protein structure are in the allowed region, are in the generously allowed region, are in the disallowed region, and hence are outliers. So if you find that a large number of amino acids are in the outlier segment, that means they are not in the proper conformation. They do not have proper phi-psi angles. So your structure would have to be re-evaluated and regenerated. But if you find that yes, the the amino acids are in the allowable limit, then you can use that structure for subsequent analysis. So what kind of subsequent analysis can you do when you have generated a structure? Remember, briefly, Professor Bandopadhyay talked about docking. And when we talk about docking, we are talking about how two molecules are interacting, whether the molecules are comfortable with each other when they are interacting. Now, these two molecules may be protein-protein, these two molecules might be DNA-protein, these two molecules might be protein small molecule. So any kind of protein metabolite, anything. So two molecules, two biomolecules interacting together. Now, when two biomolecules are interacting together, both have to be comfortable with each other, just like Kelvin and Hobbes are comfortable with each other. So you have to consider, whenever we are talking about docking, we are considering how comfortable two biomolecules are with each other. And there are definitely methods and processes by which we can explore that. I'm not going into the technical details since this is not a technical lecture, but there are methods by which we can evaluate that as well, which allows us to understand whether the docking has been done perfectly or not. And there are two, this is just for your knowledge, that there may be two fundamental approaches towards docking. One is the rigid body docking and the other is the flexible docking. So these are two essential approaches in case of docking. And post-docking, so this is where I said that we evaluate the docking complex that has been generated. So this is post-generation and scoring, post-generation and scoring. So you can see a molecule can interact with the other molecule in various different poses. But if you are to sit on a chair, for example, the chair that I am sitting in, I have been blabbering for one hour. So the chair has to be comfortable for me to allow me to do that. So if I'm not comfortable in the chair that I'm sitting, then I won't be able to do that. Just like a small molecule or a protein will not be comfortable with the other molecule with which it is interacting with if the measures and the poses of docking are not right. So pose evaluation and scoring of the poses following docking becomes a very important aspect to evaluate in terms of analysis. And these are just some images to make you understand that what can be the different poses. So you can see that in some of the images, the small molecules here, so this is a protein small molecule interaction and or protein metabolite interaction. And as you can see, some of the in some poses, the small molecules do not appear to be properly bound in the respective cavities of the proteins. So these kind of docking interactions won't give us proper complexes. However, complexes such as this, where the small molecule has actually gone and bound to the specific places where they are comfortable with, the docking sites or the pockets in the protein structures, would allow for more successful docking runs. So what does docking allow us to do? Why do we do docking? So you might be thinking about, why do we do docking? We do docking because we want to contribute towards the field of drug design and drug discovery under the umbrella term computer-aided drug discovery, CADD. And I'm not going into this detail because probably vaccine design has been hitting us since 1.5 years. So we already know how it takes clinical trials. So all these terms have been discussed over and over again over the past 1.5 years. But we need to know the concept of virtual screening, because this is one of the fundamental aspects of your computer-aided drug design process. And here you start with a compound library, which is your database, again, a database of compounds. And you are trying to find out one or two compounds which will probably serve as candidate drugs. So you have to bring down that number from 1 lakh to 2 or 1. As you can see, the library of compounds to one new chemical entity, NCE, one new chemical entity. So this entire process can be done computationally. And this is referred to as virtual screening. So what are the what are the two important aspects of virtual screening? Again, I'll just give you some keywords which you can look into later on if you are interested. So one is your Lipinski's rule of 5. This ensures the bioavailability of a particular small molecule, whether the small molecule is bioavailable in the body. And a molecular weight of less than 500 Daltons allows you to understand whether that small molecule which you are exploring has the potential to cross the blood-brain barrier. So anything which is greater than 500 would not cross the blood-brain barrier. So that is one of the components of your Lipinski's rule of 5. Though, interestingly, though it is known as rule of 5, there are only 4 sets of rules. But all the 4 sets of rules deals with numbers which are divisible by 5, and hence it is known as Lipinski's rule of 5. So number of hydrogen bond donors can be less than equal to 5. Number of hydrogen bond acceptors can be less than equal to 10. And the octanol partition coefficient or the lipophilicity of a compound should be, the log p value should be less than 5. Once we have established the bioavailability of a compound, then we evaluate the ADMET properties. So the ADMET properties are the absorption, digestion, metabolism, excretion, and toxicity. So ADMET, how much the compound is being absorbed in your body, how much of it is being utilized through your digestive tract, how much it is being metabolized into other accessory compounds, how much of it is getting excreted, and whatever is being retained, how much is the toxicity of that compound in the body. So Lipinski's rule of 5 and ADMET evaluation allows for the successful prediction of a new chemical entity, which can then be evaluated using studies such as this. For example, this is a study with HIV-1 protease. You all know regarding HIV and AIDS, the disease it causes AIDS. So this is in short a study with HIV-1 protease, which studies the interactions of various small molecules and natural compounds with the protease structure. So you can see this is the protease structure, and these are the different small molecules which were evaluated for their binding efficacy to the protease. And we found that these were so ritonavir, saquinavir, indinavir, nelfinavir. These were compounds which were established drugs which were used to treat HIV infection. And we performed a set of analysis where we utilize a set of natural compounds, plant-derived compounds, and we evaluated them and compared them with the efficiency of the others. And we were able to predict that conduragnamine B appears to be a better inhibitor of wild-type HIV-1 protease than 2 established natural compounds. So this is just to give you an idea regarding how CADD can be approached using computational methods of drug design and discovery. So there are many facets which can be discussed, but I'm not going into the details since we have already probably overshot the time. 2 things just I want to add to whatever I have been saying, that you can also think about becoming a contributor to the field of bioinformatics by developing databases. So that is one of the things that is very important, because how you are establishing repositories for other people to access and evaluate and compare their data from. So database development with biological data is also a very important sort of research avenue which people coming in from into bioinformatics can explore. Whether you can design a database which allows you to which allows the user to explore the database and then compare his or her own data. So these are some of the databases that my group has developed over the years. So this is a database which is dedicated to West Bengal butterflies. We have been able to generate a database on ferns and fern metagenomes from in the Indian Sundarbans. And currently we are working with this antisense oligonucleotide database, which is expected to be launched in October before the pujas, where we have worked with antisense oligonucleotides and how they can be designed against pathogen genomes. So that gives you an idea regarding the regarding how databases can be developed as well. So what are the skills that are required? And these 4 images I generated yesterday night itself to give you an idea regarding the nature of bioinformatics and the different aspects of bioinformatics that are prevalent all around the world currently. These are generated using Google Analytics. And the keyword bioinformatics jobs gives you a hit % ratio such as this, where you can see that a large number of keywords pertaining to bioinformatics jobs is concentrated in these countries. If you come down to next generation sequencing, which is a burgeoning field in computational biology and bioinformatics, you find that a large number of countries are dealing with next generation sequencing and experiments such as this. 2 programming languages which are being widely used, Python and R. So Python and R are being used globally, as you can see. Only the uninhabited areas are remaining unaffected by the Google Analytics pipeline. All the other areas are showing a large hit % ratio in terms of the programming languages that are being used. So you can understand that if you are actually coming into the field of bioinformatics, not only do you require knowledge of biology, which is very important, you need to have the ability to connect the dots, and you need to have some basic skills in programming, either in R or in C or in C++, and you can take training and develop it. When we started learning bioinformatics, and that was way back in 2002, 2003, we were also exposed to the initial programming languages, C, C++. Then we gradually developed our skills in R, Python, Perl, BioPerl, etc., which have come over the years. It doesn't happen in an instance. You do need to be patient. You need to be perseverant in your efforts towards developing those skills. But you have to have an idea regarding what needs to be there. And bioinformatics offers you the opportunity to explore both academia as well as industry. And this is just a graphic to give you an idea regarding what kind of analytics are used mostly in academia and what kind of analytics are used mostly in industry. So anything which suits you, you can, based on that, you can either get into academics or you can get into industry. So that is something which you have to keep in mind when you are actually getting into the field of bioinformatics. So this has been my team, which has been with me. Majority of them are still with me since 2006. Currently, these are some of the collaborations. The past 1.5 years have been very good in terms of the contributions that we have been able to make. And I leave you all with this particular quote from Craig Venter. And Craig Venter is a very interesting personality in terms of your human genome project. And you can take a look at the contributions of Venter itself. So I leave you with this quote that we are clearly much, much more than the sum total of our genes, just as our society is greater than the sum total of each of us. Our physiology is based on complex and seemingly infinite interactions amongst all our genes and the environment, just as our civilization is based on the interactions amongst all of us. We are confined in having interactions over this digital platform. And I end with the hope that very soon we will all return and we will come out of this digital divide and we will meet in person. Thank you very much.
Thank you, sir. Thank you very much for your detailed and wonderful talk. Am I audible?
Yes. Thank you.
Really, your explanation provided deep insight in how to deal with a particular case and study it very sequentially. I think this would definitely enlighten our students, and it was really very, very lucid and very wonderful presentation. Now I would request my colleague and joint convener, Dr. Shumanti Gupta, to conduct the question answer session.
Yeah, am I audible?
Yes.
Yeah, you are.
Yeah, thank you, Dr. Shelley, for offering me the podium to conduct the question answer session. But before I start with my session, I would like to take the opportunity to thank both of our respected speakers, Professor RAJIB and Dr. Sayak Ganguly, for consenting to act as resource person of today's event. Especially to both of you for your lucid and detailed talk. Especially Sayak, your talk was literally mesmerizing. I hope our students have acquired appropriate information about this branch of growing science and also gained adequate motivation to pursue their studies on bioinformatics in future. So now to deal with a few questions, very specific and very
Shumanti?
questions.
Hello, Shumanti?
Yes.
I think your mic is causing some disturbance and your video is not visible. Please put on the camera. দেখা যাচ্ছে না তোমাকে।
Yeah, is it okay now?
Ha ha ha. Yes.
Okay.
Yes.
Could everybody hear what I said? What I've said, or
Yes. It was audible, but some disturbances were there. So I
think
okay, you can continue. Huh.
Okay, anyway, sorry for the interruption. So to deal with a very few questions from the students' perspective, already you have said regarding the programming necessary for a student of bioinformatics, but if I ask you to provide a hierarchy, what would be the initial program if a student starting as a beginner? What would you suggest with the type of program he should learn and then go on for higher programming?
Basically, every programming language is based on a programming library. So suppose if you are looking at the programming library that R as a programming language uses, the programming library of R is written in C. So basically, as a first step, it is always better if we learn C and C++, which gives us an idea regarding the data structure of programming. So first step should ideally be C and C++, and then progress further in whatever directions the student requires.
Okay. My second question is, like sir was already talking about using mobile phones for bioinformatic analysis. How much do you think should be the minimum RAM speed or the minimum storage area for a mobile if a student wants to initially start his bioinformatic journey with the mobile phone itself?
Now, I'll answer it in two parts. The first thing is that mobile usage will have certain restrictions. For example, we will not be able to install certain software in the mobile phones which allows for bioinformatics analysis. So our analysis has to be web-based. So, and there are several tools web-based which are available. So there is nothing to feel that if you are unable to install software, then you will not be able to perform analysis. That is not the case. But since it is a web-based analysis, more than the RAM, the connectivity would be an important thing to look into, and the kind of data usage that the student can afford in terms of downloading and uploading of the information that has to be exchanged over the web. And even if no storage space, if there is low storage space in the mobile, the student can have a Gmail account which allows for 15 GB storage space, a separate Gmail account, in which he or she can use Google Drive, where he or she can store the data and then access it as and when it is required. So basically, if you are really looking for specific, then 4 GB for 3 to 4 GB RAM mobile phone with 32 GB storage should suffice.
The next part is, as we are all passing through the pandemic, a question which is still related, although there are lots of vaccines which have already come up, but still, what do you feel, what more the bioinformaticians can do to get rid of this pandemic ahead?
I think bioinformaticians can do only or give us idea regarding the data that is being presented to us through the various genome sequencing consortium projects that are coming up. So basically, we are in a position to comment on the contributions that the mutations are making, and whatever data regarding mutations are being talked about in the public forum are all as a result of the analysis that bioinformaticians are doing at the back end. So whether a particular mutation is more lethal, whether a mutation is making the virus more transmissible, whether certain mutations have the potential to infect a particular group of people or from different ethnicities. All these data are being generated and are being contributed in the scientific forum, which are gradually coming out into the media and into the public space as well. But typically, to get rid of the pandemic, I think we have to wear masks and be safe.
Very pertinently said. The next question is, how much precision is expected from the dry lab experiments or data that would help in designing the wet lab experiments? Now, I'm talking about the precision.
Right. So this is a very important question, and this is a point which probably is a point which requires a bit of experience with dry lab analysis, because if we are submitting data to a server, then definitely that server will be giving us some kind of a result. Now, it is up to the user to understand the result that has been obtained through the analysis of a server, whether that result is pertinent to the kind of analysis that individual was planning to do, or is relevant in terms of the analysis that was planned in the first place. So this is exactly the point in terms of precision. This is probably the only aspect and probably one of the most important aspects for research personnel, where you do require some idea regarding what kind of results you expect, and a more biological insight towards a bioinformatics issue would help in the understanding of the result and thus increase the efficacy of the design.
Yes. The next question is, I would like to combine two questions. What do you think is the future of this growing study? Or in other words, what do you think are still the limitation areas of this branch of science? I think the answer will be a common one.
So actually the future is fantastic, because why I say future is fantastic, because biological data and the nature of biological data and the variety of biological data will not cease to be generated. So we in the future also we will be getting data pertaining to different biological experiments, more and more fields. For example, I have not talked about a field which has greatly been contributed to by medical computational biologists. So computational biologists who are working dedicatedly in the medical field, and that is image analysis. So image analysis has become a fantastic area to work with. What kind, see all the pathological interventions that we go through. I just talked about the CT scan very briefly. So CT scan, MRI, etc. All the images that are getting generated in terms of human diseases, human physiological diseases, physiological conditions for evaluation of human subjects. Image analysis has become a very important aspect to work on, and that also requires a bit of programming language, that also requires insights into biology, insights into medical science, and together only people who are able to merge these two together are getting successful in the field. So this entire field and next generation sequencing is evolving day in and day out. Every 2 years we come up with a new technique. So as soon as a new technique comes up, then obviously the data analytics changes, the algorithms change, the algorithms have to be reworked. So there is constantly a sea of change that is happening, and as sea of changes happen, opportunities also increase.
That's very rightly said. And one final question. This is from my point of view, like going through the talk, your talk, hearing it was, I was like mesmerized, as I've said. So this is one question, I have my experience with a very brief part of bioinformatics. But now that one thing we have to know that we have lot amount of genomics data, transcriptomics data, metabolomics, metagenomics, all the data. But ultimately what we are more looking into this whole how this biome world totally interacts. So can we dream of anything what we call interaction database, which would incorporate all the interactions made by this entire biome world, what we call the interaction database. In the long run, do you see that in the long run this thing also can be done? Like NCBI, this is a very basic database where we get all the genetics that is shared throughout also. So in future we will come to know about the interaction about all the biome world by looking into one interaction database. Like this is a very
that's a very interesting. I just give you an insight into this because very recently I was inducted into the Mangrove Microbiome Project initiative, which is taking place all around the world. So it is a contribution of 10 laboratories around the world, which is contributing to the Mangrove Microbiome Initiative. And there, the mangrove areas which are interspersed all around the world are being studied. I am just working with the Indian Sundarbans briefly. So there, this holistic data coverage of the genome, the transcriptome, the metabolome, and the microbiome taken together as an organismic system. So this is what is being planned, and definitely once the data gets generated, at least this will yield a database at least pertaining to mangroves all around the world, which will house such information. So for example, we recently had the genome sequence of Abyssinia, which was published very recently, a few days back. So there also we will be able to understand the functioning of the genome, the transcriptome, the microbiome, and how each of them is contributing towards the other. Because now we are in a position to definitely say that there is a continuum that is there. So this continuum can only be explored, as you rightly said, in terms of the understanding and exploration of data and contribution and development of databases, which deals with all kind of data corresponding to a specific organism. So you take an organism, you evaluate it completely, and you contribute the data. So that will definitely be the future of bioinformatics research, definitely, and sequencing based research, definitely.
And so we all look forward to that.
Yes.
Okay, that was all for the question answer part. Thank you very much for the very, very point wise and very detailed answer. And I don't think so we have Dr. Bandopadhyay with us. Dr. Shelly, do we have Dr. Bandopadhyay with us? Okay. I would like to ask him 2 questions if he is available.
Yes.
Thank you, sir. Thank you for your very lucid talk and very student-friendly talk you had provided. So, a minimum, I had already shared the question with Dr. SAYAK. I thought you were not there. So what would be, again, the basic questions from the student's point of view? What do you think to start with a basic smartphone? How much minimum RAM speed and minimum space do you suggest for the beginners to have in their mobile phone so that they can initiate the study of bioinformatics?
Actually, in any smartphone, you can start with, because you will not download any software or you will not store anything. Just for visualization at the UG level, you can see the different databases. You can perform some experiment based on online available free software. So any kind of smartphone it can be used. When it comes with the downloading of the softwares or molecular simulation, docking, then only you need the help of computers. But the minimum experiment, what I saw and what is designed at the UG level by UGC, that is possible by any kind of smartphone. which are normally students use.
Okay, sir. I got your point. So thank you very much. Thank you. I will thank both the speakers for their talks. And now, as we have already reached towards the completion of our program, so Dr. Shelley, with your due permission, would I continue with the end of the session?
Yes, yes, you continue. You may continue.
Okay. Today's talk has not only enriched our knowledge, but I hope has also helped in eliminating many knowledge gaps as well. So on behalf of my entire Department of Botany, I want to thank our notable speakers, Professor RAJIB and Dr. SAYAK Ganguli, for their informative and very much pertinent talks, especially going by the time we all are passing through. This confined situation due to COVID could indeed be rightly used by the young students for learning different aspects of bioinformatics. Besides, I'd also like to extend my sincere thanks to our principal, Sir Professor Prashanta Bhattacharya, for his steady encouragement in conducting these types of talks. We are also grateful to our IQAC coordinator, Sir Professor Tanmoy Bandopadhyay, for his invaluable advice and guidance from behind. Besides, I also thank the conveners of IT Subcommittee, Dr. Rabiul Alam and Dr. Shubhajit Mondol, convener of Academic Subcommittee, Dr. Sucheta Joy, for their wholehearted support. I would specially thank my joint conveners, Dr. Shelley Sinha and Dr. Joyita Shaha, our deputy convener, Mr. Shagatu Mukherjee, for constantly putting their efforts in making this event successful. I would also like to thank my faculty colleague, Mr. Arju Dash, our laboratory instructor, Mr. Sharbashish Banerjee, our lab personnel, Mr. Devabrato Pandit and Mrs. Shubrata Adok, for providing their mental support from behind. Last but not the least, I thank all the participants and all their students for their participation and appreciation, without whose support this event would not be successful. So thank you all. I end with the saying, stay home, stay safe. Thank you.
Thank you.
Thank you.
We'll stop the recording. The live stream.
The recording should be stopped and we'll leave, isn't it?